we basically want to sequence the maize genome and thousands of maize genome to identify the haplotype diversity in the maize population. So today, with all the sequencing technology we have, and the major challenge of sequence most of genome is not technical, but how do we can sequence the genome cheaply and with lower cost. So in the crop genomics world, so the strategy we have been taking is to sequence genome at 3 levels. Level 1 is to do de novo genome assembly to construct the reference genome. So reference genome will be used as a coordinate point to anchor all the genetic variations. The second level is we're doing whole genome sequencing to capture the major diversity in the haplotypes. Then when we sequence each individual, once we have the reference genome and we have a haplotype map to represent the genetic diversity in the population, then we come to each individual genome. We can just do very sparse sequencing at a very low cost. The purpose of doing this is just identify boundary of haplotypes. Then we can fill in the boundary with the haplotype which captures in the haplotype map. Then by this, we can deliver genome very cheaply. So in this presentation, I will just talk about how we construct the maize haplotype map, and especially now we are in the transition from using the single reference B73, a single genome B73 as a reference genome, to using the pan-genome as a reference. So I'm talking about our strategy, how to first using single reference and to using the pan-genome reference. So when we use single reference genome, we had to heavily rely on the genetics of the population genetics information in the population genetic information to filter the false positive markers. But when switch to the pan-reference genome, we can do because we have a genome of all the represent the major structure variations, we can rely on the alignment of physical genomes to identify good markers versus bad markers. So we basically try to, using the new approach, try to be able to sequence a new genome cheaply, that can be more practical, used for the plant breeding practice.